and I'm going to hand over to Dr. Karteris.
Thank you very much. Right, I'm Manos Karteris. For those of you who don't know me, I am a reader at Biosciences. And where I would like to take it from is that we talked about the process of fertilization. We talked about the blastocyst. We heard about what sort of vitamins are really important for the maintenance of pregnancy. And I would like to take you in an interim in a key transient organ that is really important for the maintenance of pregnancy, and that is the human placenta. And if we look historically, Leonardo da Vinci nicely sort of drew the famous fetus in the womb back in 1511. But since then, a lot of research has gone into to understand a lot more about pregnancy and what's happening in utero with the right use of techniques and apparatuses. We can now have a 3D scans of babies. This is my daughter back in 2004. And of course, a lot more work has gone to understand what's been happening during these 9 months of gestation. How these 9 months in the uterus, how these 9 months in the womb really act as a hardwiring process for later on in adult life. So Joe spoke about the blastocyst implantation, and essentially what happens is when we are reaching a 32 to 64 cell embryo, it starts containing some distinct sort of differentiated cell types. And essentially the outer layer, being the trophoblasts and the syncytiotrophoblasts, these are the cells that go on to form the human placenta. The inner mass that you see here goes on to form the actual fetus and the fetal membranes. Now, how many of you have actually seen a placenta or touched a placenta? Okay. I can see a few dads raising their hands in the audience. Right, okay, so the human placenta is a discoid organ, is about 25 centimeters in diameter, about 3 centimeters thick. It's quite spongy to feel, right? And it weighs about 0.5 kilogram. It's normally attached to the upper part of the uterus. What else? Inside the placenta, if we concentrate on this kind of diagram here, we have the fetal villus trees. And it is these fetal villus trees really that are bathed directly with maternal blood. And it is at that area where we have the exchange of nutrients, the exchange of gases, the exchange of hormones. And the placental development starts about 6 weeks of gestation and is completed around the 12th week. And of course, we have the umbilical cord here that is normally attached to the center of the placenta. Right, so the placenta does a lot of functions. We can spend hours and hours. I'm cognizant of the time, by the way, so I think we'll just sort of outline some of the key metabolic and hormonal functions of the human placenta. So what does it do? Okay, well, there's a lot of things. It can act by being an organ where gas transfer takes place. The mother passes oxygen and the fetus passes CO2 back to the mother. It is also an organ that we see waste removal from the baby. One of the main functions in terms of metabolism is that of providing some initial immunological support by passing immunoglobulin G antibodies to the fetus, but also being the organ that delivers key nutrients. Nutrients as glucose, such as amino acids, the building blocks of proteins. These are really essential. essential for fetal growth and fetal metabolism. It also passes soluble vitamins like B vitamins, beta vitamins, and also fatty acids. However, not everything is rosy. In the human placenta can also be an organ that certain viruses and drugs can also be passed from the mother to the fetus. Things like viruses, like HIV. Or more recently, I'm sure you all heard about the Zika virus, right? So again, with the Zika virus, it can cause serious problems during pregnancy. If a pregnant woman is infected with the Zika virus, it can be passed to the baby. It can cause some serious problems, and one of them is that the baby is born with what is called microcephaly, where the head size is much, much smaller. We can have drug transfer. Again, that depends on the type of the drug and the way it's metabolized. But again, to give you an example, almost all the anesthetic drugs can really cross the placenta. Or we have some other areas such as toxoplasmosis, for example, that can cause problems during pregnancy, including miscarriage, preterm birth, or stillbirth. So something to be cognizant, something to be aware. In terms of hormone secretion, again, the human placenta is what I call a transient endocrine organ. It secretes a wide repertoire of hormones. Some of the main ones is estrogen and progesterone. They play they can interact each other in the sense that initially progesterone maintains a quiescence in the uterus. Okay? It suppresses smooth muscle contractility, allow for the fetus to grow. Later on, as gestation progresses, estrogen levels increase, and by doing so, we have a shift from a contractile state from a quiescent state, where everything is relaxed, to a contractile one, allowing the onset of pregnancy. And this works in conjunction with a number of different hormones, for example, relaxin, that relaxes the pelvic ligaments and softens the cervix at the end of the pregnancy. Other hormones includes the human placental lactogen or human chorionic somatomamotropin, as else known. Again, a key hormone that breaks down fats, is involved in what is called lipolysis, also freeing up glucose for the fetus in a process called gluconeogenesis. Other hormones, corticotropic releasing hormone, a hormone that is identical to the CRH that is released from the human hypothalamus. And again, it gives you an idea of how a transient endocrine organ, as I earlier said, placenta is. There are studies that suggest that CRH can indeed establish the timing of birth, but more importantly, CRH drives fetal organ maturation, like fetal lung maturation in particular, as well as there is some evidence that it might also affect the placental vascular tone. And as I said, these are just a few of the hormones that the human placenta secretes, along, of course, with the very, very first one, the human chorionic gonadotrophin, the one that really, HCG really elevates at the very, very early stages of pregnancy. Indeed, is the hormone that has been picked up in the pregnancy test. Other hormones include adiponectin and leptin. Hormones that are secreted, initially thought are only secreted from fat cells, but actually the human placenta does secrete them. Again, underpinning the importance of human placenta in the fetal sort of metabolism. Again, there is a lot of things that can go wrong, and one of them is pre-eclampsia, where we have some defective placental formation. Now, pre-eclampsia is defined as high blood pressure during pregnancy, and essentially the human placenta doesn't get enough blood. And you can see here the difference between a normal placenta and pre-eclamptic placenta. And the problem is, but by having a defective or compromised placenta in the structure, inevitably the baby will start receiving less oxygen, fewer nutrients, and of course that can impact in fetal development. So subsequently, pre-eclampsia can be an important contributor to preterm delivery. So the question is, how do we study placentation? How do we study how the placenta works in health and disease? How can we do this? Well, believe it or not, to date, modeling the physiology of human placenta still remains a challenge, despite great scientific advancements made in the field. And I'm sure you can all appreciate here that animal models, although they are very useful, cannot really fully replicate the structure and function of the human placenta. The mouse, for example, has a gestation period of 21 days. Different hormonal milieu during that time. There's no way you can recapitulate what's happening during 9 months of gestation. Moreover, there is animal welfare legislation worldwide, encouraging, and rightly so, the use of 3 R's: the reduction, refinement, and replacement of animals. Okay? So we really have to take all this into consideration. So the question is, what efforts have been made to develop alternative models for the placenta so far? Well, we can start with cells, right? I'm sure all of you are aware of cell cultures. This is nothing new. The cell cultures have been around since the early 1900s. And as I've put here, what has been a very useful model is cells growing on a monolayer, or as you can see here, into dimension. They are inexpensive, easily accessible, well established, and there is a lot of comparative literature using 2D cultures. However, the issue remains that it is what it is here. It's a two-dimensional. And let's face it, all our organs are not 2D, right? All our organs exist on a 3D structure. So we are now started moving into 3D cultures, since they provide a better way of representing human tissue outside the body. And essentially what 3D cultures are is that we give the cells an artificial structure, if you like, an artificial scaffold, where cells can grow into spheroids or into clusters, resembling much more a 3D image. And to give you an example of what's happening at Brunel, this is a model that we have used for placental cells. These are called bio cells. And you can see here the cells on the 2D. We see the nucleus here stayed in blue. We have the cells, the cell membrane stayed in green, and this here when they're merged as an image. And when we grow them into a nitrocellulose sort of scaffold, this is how they grow into 6 days. They start forming these beautiful spheroids around 7 days, and this is how it looks. Again, you can see here you have a cluster of cells. Again, the cell membrane is staying in green, and here we have sort of the merged image. These cells, again, have been grown into what is called a wood-based nanocellulose hydrogel. Again, an animal-free derivative, really driving further what I've talked earlier on about the 3 R's. And in collaboration with Tampere University up in Finland, we set up these cultures in this hydrogel, and then we have taken a 360 video of them. And you can see here the scaffold, quite opaque in color. And here you can see some of the spheroids that are formed. as they come into focus. For example, we'll look at this one and this one later on. So essentially, 3D cultures show a higher degree of structural integrity. So where are we taking this now? As part of the CenGEM, we have the Organ on a Chip group. It's an interdisciplinary team of engineers, bioinformaticians, bioscientists, and all of us came together interested in applying OOC systems to study women's health. Essentially, this organ on a chip technology brings together, if you like, two distinct fields. The field of microfluidic engineering, along with that of cell and tissue biology. So to summarize and show some of the ongoing work, we have managed to grow cells on spheroids, on clusters, using a number of different scaffolds. And the next step will be to use a simple, what we call placenta on a chip model, where we can put different cell types together, separated by a semi-permeable membrane. And essentially what we can do with these is we can put human trophoblasts, placental trophoblasts on the one layer, and endothelial cells, cells that they can mimic, if you like, the umbilical cord, and therefore start having an organ on a chip that can resemble the fetoplacental unit. Okay, so I will leave you with this, and I would like to thank you for your attendance.